Okay, so we're going to be talking about data for your research organism. I'm Peter Cooper of Adelaide Rhodes, and I are doing teamwork on this one. So I'm going to introduce sort of the topic, and she's going to take over to show you how we can get these data together in a Jupyter notebook after I go over a few things preliminarily. Okay, so today we decided to focus on this organism. This is a tree species, the black cottonwood, Populus trichocarpa. It's a fast-growing forest tree that's native to Western North America. It's used in pulp and lumber. It has a pretty compact genome, which is kind of useful, 450 million base pairs or so. And, although we say this is kind of for non-model organisms, this is a potential model tree genome. So here's what we're going to do today. We're going to basically explore and retrieve data for this organism. We're going to look at the genome assembly. We're going to gather some gene information, and we're going to particularly look at a suite of genes called the NAC domain-containing transcription regulators. These are important for plant development, and so if you were interested in conditions and how they affect gene expression, these would be genes that you'd be interested in looking at. In terms of gene expression, we're going to look at some expression data in the form of RNA-Seq, so that's next-gen sequencing reads from SRA, and we'll ultimately wind up looking at the effect of drought response on gene expression. Once we collect all that information, then you would need to analyze that. And Adelaide is going to do a demo using command line tools that she's got into a workflow setup in a Jupyter notebook. And she will analyze these data, and we'll put them in a genome browser for you to analyze and see the expression data mapped across the genome. So just to introduce that topic and to show you, to introduce some of the resources, I'm going to do some of this on the web. And on the web, we're going to visit the assembly database and the Datasets, which is a tool associated with assemblies. We'll use that to get the genome assembly and annotation. We're going to use the gene database to get the genes that we're interested in, find their locations in particular, because that's what we're going to do with them at the end. And we'll go to SRA, and we'll get the submitted next-gen sequencing data. The RNA-Seq data is what we're going to be looking at today. And we'll use a tool associated with SRA called the Run Selector to help to sort through the kinds of sequencing reads that we have there to get the ones that are of interest. And then finally, we're going to use the genome browser, genome data viewer, to load the data at the end of the talk to see the analysis that we've done in the context of the genome. And so to do that analysis, we're going to use command line tools. We're going to use the command line version of Datasets to download the data in the Jupyter notebook. We're going to use the SRA toolkit to access the RNA-Seq data. And we're going to use Magic Blast to align the RNA-Seq data to the genome so we can see the kinds of expression changes that we see over the course of the experiments. And the last command line tool, which isn't really an NCBI tool, but it's called SAM tools. And that's something that you're going to need to, if you're going to need to have installed if you're going to work with alignment files like the kind you're going to get from Magic Blast. Okay, so with that, what I'm going to do is go over to the NCBI website, and that's the URL there. And I'm just going to run through a quick demo on the website about how to get some of these data. And then we'll switch over to the Jupyter notebook and Adelaide. And I'll go to the NCBI homepage here. And the first thing I'm going to do is just do a search for the name of the organism, and I'm going to do what's called an all databases search. This is a way to search all NCBI databases at once. We've put some convenient search sort of interrupters in there. So if you search for something like the name of an organism, we have a pop-up that shows up and says, hey, it looks like you're looking for the name of an organism. Here's a way to get the information about that organism, and here's a way to get some data. So what I get here is a hit to the taxonomy database. That's where you could go to get some information about how we classify this organism at NCBI. And you can link out from there to all the different resources where we have data for this organism. We also have some shortcuts here that let you get some of that data directly without having to go there. So we're going to take a look at two of these. Let's take a look at this one where it says browse and download genomes for Populus trichocarpa. And that's going to take me to Datasets. This is the Datasets genome browser. And you can see that it's got 3 different genome assemblies for you here. The one at the top is the reference genome, the NCBI reference genome with its annotation. And we can go ahead and download that if we want. I'll just mention what these other two are. The one at the bottom is the GenBank submitted genome assembly, and the one at the middle is the previous RefSeq assembly. So we take the GenBank submitted assembly and we annotate it ourselves. So if we go here and we download this, let me check that box there, and I'll go to the download button up here on the right-hand side. I can get all different aspects of this genome. I can get the genomic FASTA, two different sort of feature files which show me how the thing is annotated. I can get the transcripts and the protein sequences. These are all based on NCBI's eukaryotic genome annotation pipeline. For the purposes of today, let's go ahead and download the genomic FASTA. So that's going to give me a FASTA sequence for each one of the chromosomes. And then I can get this GFF3 file, which gives me the annotated features. So if I want to, I can name my file. I can press this download link here, and that will just download a package for me with all that data in it. Just press it for a second so you can see that I could just download this directly if I wanted to. And it gives me a zip file that has all that data in it. So that's one of the things we're going to need to do our analysis today. I'm going to go back to get back to the sensor, and I'm going to jump ahead. In a way, I'm jumping to the end of the demonstration today, but I just want you to see this. So if I wanted to look at this genome in the Genome Data Viewer, which is our genome browser, I could just follow this link here. And because I've looked at this gene before, the Genome Data Viewer remembers what I did. And this actually happens to be one of the genes that we're going to be looking at today. This is one of these genes that has that domain in it. And if I want to, I can go to gene here. Before I do that, and that's what I'm going to do, I just want to point out to you that we already have aligned some next-gen sequencing data to this. And these are the data that we use from SRA to annotate this genome. So we use these RNA-Seq data here to get some gene models for this, and that's basically how we annotate this particular gene. So with that in mind, let's go ahead and move to gene now. And of course, we'll come back to this view later on when we align our own next-gen data that we found to the genome. Let's go ahead and go over here to gene. So I'm just going to click through to the gene record. And so as promised, this is one of these NAC domain-containing protein genes. And this is a typical gene record for an organism where we don't have a lot of information. If this was a human gene, we'd have a lot more things linked in here. This is sort of the one-stop shop. If you know what gene you're looking for, you would come here to find out more information about it. We can also use gene to get all the genes for the black cottonwood or a particular set of genes. So let's just do that search that we did before right here in the gene database. And I get my pop-up again with my ad, but I also can see the number of genes that we have for black cottonwood here. Below there's 37,273 genes. I can filter these down to get a set that I might be interested in. So I can click on this genomic filter here. That means that these are genes that are these are chromosomal genes, they're not things that are associated with the organelles. And I might be interested only in protein coding genes. I can do that here. And so now I have 30,000 genes, about the same number as the protein coding genes for the human genome. And I could add something to my search if I want to. I could add that NAC domain search if I want to. Let's see what we get if we do that. So I could make this more precise if I wanted to. I could say NAC domain containing protein, and that might even be more precise in this, but it looks like I'm getting a lot of things that are of interest here. And let's narrow this set down. There's 116 of these genes with that kind of description. Let's narrow this down a little bit more. Let's just get all these kinds of genes that are on chromosome 5. And what we'll do when we align these things, we'll particularly look at chromosome 5 just to get us a smaller set of data. So here's another filter that I can add. And let's add chromosome locations. Let me show that filter. I'll go ahead and click more. And I'll click on black cottonwood. It doesn't matter which one of these I pick. That's the common name. That's the scientific name. Let's pick chromosome 5. Let me apply that. And it takes a minute. And so I get 10 of these genes on chromosome 5. So if we wanted to browse these across the genome in the genome data viewer, which we will do later on, we could look at perhaps changes in the gene expression of these genes in different treatments of some kind of experiment with RNA-Seq. So what we want to do now is to find the RNA-Seq experiments that we're going to work with. And so I'm going to change databases here. Let me just take that NAC domain off of there. Let me change the database to SRA. SRA stands for the Sequence Read Archive, and that's our repository of next-gen sequencing data. I'll go ahead and run the search there. And I get 2,700 experiments, basically. And most of these experiments are one sequencing run. Sometimes there's more than one. I know that I'm interested in expressed sequences, so I can use the filter over here to get RNA. So I now have 1,800 RNA-Seq experiments. And I could, click on every one of these and read through what the experiment was and all that stuff, but it's kind of awkward to do in this way. And it's also true that because these are submitted data, the kinds of information that people provide to us varies a lot. And so it's kind of hard to sort through them in this interface. There's a better way to work with these data, and that's the Run Selector. And so I'm going to throw these all into the Run Selector, which is a good way of looking at the various kinds of data, metadata that people provide about their experiments. And then I can use that to sort of sort down to the experiments that I want to look at. Okay. So here they all are. And there's a table down here that has all these different columns. Center name, you can see them here. It goes on for quite a ways. Sometimes there's nothing in that column. They didn't give us that information. You can scroll across and see there's a whole lot of potential information here. Sometimes it's filled out, sometimes it's not. And I can actually select by those columns to see how we can split the data up into various things. So let's just look at the various columns that we could have here. So for example, here's a simple one. I can see what country the geographic location was listed. So we have Canada, uncalculated, China, and USA. And most of them don't have that information on them. More relevant to what we're trying to do today might be something like drought treatments or things like that. So here's an interesting set, days after drought treatment. Let me check that. And there are 3 different treatments, 0-day, 5-day, and 7-day. So I can go ahead and ask to see all those. So now I have a set of data that might be interesting to work with. We have 4 sets of samples from each of the treatments. And I might want to look at the expression of some of those genes that we saw a few minutes ago at these different days of drought treatment. Now, let me go ahead and select all these. Now, how could I get these data? Well, if I'm working in a situation where I can get them delivered, I could get them delivered to the cloud if I want to, or I could get them delivered to a Galaxy workflow. What we're going to do is to get them with the toolkit, and in order to do that, we need to get all we really need to do that is to get the accession numbers. So there's only 12 of them. If I had hundreds of them, it might be particularly useful to download it. And notice that I can download this entire table, or I can download the accession list. And if I click here, all I basically get is a list of a text file that has those 12 accession numbers in them. And Adelaide's going to use that information in a few minutes to align these to the genome. Okay, so let me switch back to my slides. because we're going to shift gears now, and we're going to use these data in a Jupyter notebook in a workflow. And so basically what's going to happen is we're going to use the same sets of data that I just showed you, but we're going to incorporate them into this workflow. So we're going to download those SRA runs, and we're going to use the SRA toolkit and a function in there, or a program in there called FastQDump, to get that data out of SRA format. We're also going to download the assembly using the Datasets command line tool. And then we're going to use a tool called Magic Blast. And we'll go ahead and use Magic Blast to align those next-gen sequencing data to the genome. And that will tell us, like, what genes they match. And we'll process that alignment data with SAM tools so that we can upload it into the genome data viewer and look at expression across the genome and also across those different treatments. So what I'm going to do now is change presenter and give this to Adelaide and let her take over for a while.
Great. Can everyone hear me? Okay, great. Good. So this is a notebook. For those of you who may not be familiar with notebooks, it's just a way to show command line and let people practice on it. So we have actually provided a link to a live notebook that if you want to reproduce this analysis, you can do it from anywhere in the world, because a binder notebook will spin up a small server somewhere else. All you need is a web browser. So anyway, we're inside the notebook, and I did change the order a little bit. We're going to get the SRA runs first, and then we're going to get the assembly, and then run Magic Blast before we load stuff into the viewer. So here, these are the 4 steps we're going to follow. And then if you do choose to download this notebook, so you can get it just by hitting download here, you would need to install a few programs onto the machine where you might be running this. But basically the way Notebook works is you have just your code blocks and you hit run, and then it runs a little bit of code. So the cool thing about this is that I could write one notebook, but use it for many different analyses. So I have gone ahead and started looking for data that's related to our SRA run. So remember, we had an accession list that was output. So this was the first accession in the accession list. And I'm using the SRA toolkit tool called FastQ-dump. So this is the actual command line. So the way notebooks work also is you can run bash commands, Linux commands, by just putting an exclamation point in front of the command. So if you actually just wanted to do this on command line, you would copy this piece of the code, put it in a terminal, and you would be able to run it if you've installed SRA Toolkit. So the new SRA Toolkit, just a little bit about this code block, does require a little bit of configuration, but it's easy to do it interactively, or you can use just a little bit of code to get it started running. What the purpose of that is that we're on the cloud. NCBI has moved 18 petabytes of publicly available data that's not controlled up to AWS. So SRA Toolkit needs to know where you are and how you want to access the data. So it'll default to the free buckets in AWS. So I ran this command. I asked for the first 2 lines in a FASTQ format from this particular sequencing run. And as you can see, I'm not going to run the code cells live because they take a few minutes. So I've pre-run them, but this is the expected output. You would see the first 2 lines of the FASTQ file. All right, but I don't just want 2 lines. I want to actually do an analysis here. So what's my strategy? Well, I'm going to have to do something called batch, which means I send a whole bunch of numbers into the program to run. And so there's different ways of doing this. And, I've been doing bash for a long time, so I apologize if you're not familiar with these commands, but there's many ways to do this. So the first thing you need to do is have a list of your accessions. So this is the list that Peter downloaded. And I'm saying open it up. So cat just means open up the list and do something with it. And then you would send it to another command. So this little up and down line is called a pipe, and it sends it into the next command, where I'm saying I've got 12 of these files to run. Let's run FastQ-dump, but this is a small machine. I don't want to get all the data. I'm just doing like a preliminary survey of what's in it. So I only want 1 million lines. So I'm getting 1 million lines for each of these accessions, and I'm putting it into an output folder called data. So like I said, I've already run it. It took about 3 minutes to run this command. And so after the command is finished running, I've got a nice little data folder filled with FASTQ files that are 1 million lines each. Okay. The next stage is going to go and get the reference genome. So our datasets group at NCBI has spent a lot of time building command line and Python and R versions of their tools. So I like Bash, so all my commands in this notebook are written in Bash for the most part. And so what you can do is download and install a program, and it's called Datasets. And when you call Datasets, you can tell it to do a lot of different things, like download genes, but I would like to download a genome accession, right? So I actually have the genome accession number we were looking at in the web browser, and I only want chromosome 5, so I've already incorporated that into how I'm downloading it. And I do want the GTF file, but I don't really want the protein FASTA or the transcriptome FASTA. I just care about the annotations. And just FYI, the GFF is included automatically, so you have to exclude it. You have to opt out of that. All right, so that command runs and gives you a zipped data file. So just for my convenience, because I might be running many of these analyses, I've renamed the file upon output. So what I've got is an unzipped of the file, which came down as NCBI dataset.zip by default, and then I'm renaming it into a directory for Populus trichocarpa, and I call it RefSeq. So I remember that this is the RefSeq dataset, and then as you can see, it outputs several different files. But the one I really care about and what I'm going to use in the next step is the chromosome FASTA file. The nucleotide version of the FASTA file is what .fna means. Okay, great. So I've done the first two steps. I've gotten my FASTQ files set up. I've got my reference genome. I'm ready to align. So here's another chance to highlight an NCBI tool called Magic Blast. Magic Blast differs a little bit from other aligners in that you do not index the genome. What you actually do is make a BLAST database of your target FASTA file, the thing you want to align to. So I've done that up here. I've made a BLAST database using the BLAST Plus tools, which are loaded into my computer here, and have generated a mini BLAST database just for chromosome 5. It's nucleotide, and so now I'm going to be able to use Magic Blast or regular blast, any kind of blast. Like, it's just making a BLAST database for whatever downstream purposes I want. So how does this all work? Well, Magic Blast will take as input your reads, and you could give them directly from SRA. You don't have to make them into FASTQ files, but the reason I'm doing FASTQ files today is because they're smaller, and I know they'll run in the amount of time and memory that I have available on this little mini machine. The default output is going to be SAM files. So SAM files are really useful also, but I know that I'm going to need BAMs for loading to the genome viewer. So that most genome viewers require a BAM output to load. So I go ahead and make a little directory called BAMs. I set my machine virtual memory limits by just limiting the amount of bases analyzed at one time by Magic Blast. That's all that this tells you, but you wouldn't have to do this if you're on a larger machine. And then I start the command. So for those of you not really familiar with command line, what you get is you have a command, and then you generally have a bunch of parameters that say what data sets do I want to analyze with this command. So you usually have a query data set. So I'm just running one file here. I'm just picking one of the FASTQ files I made in the last step. I'm saying what database do I want to analyze it against. And so that would be the BLAST database we just made. And then you can set other parameters such as splice, let it be true because it is transcriptomic data. I say no unaligned just to save space because the SAM file won't retain reads that weren't aligned. And then I have to tell it it's a FASTQ file. And then once again, I'm trying to conserve virtual memory on the small machine, but you wouldn't have to set that parameter there, the max DB count. All this information is available in the links up at the top of this notebook. So you'll be able to do more exploration on how all this works later on. And then, as I mentioned, we need to use SAM tools to convert it into BAM. So I go ahead and use the little pipe tool again to start sending the data that's made by Magic Blast into a BAM file. And then after I'm done, I just have to index it. And then from command line, I've never had to go back to the web or look at anything on the web except maybe getting the accession number for the reference genome and getting the accession numbers for Run Selector. I've been able to do all of this in the notebook. So now I've got a BAM and an index BAM And that brings us back to the web, where we're able to view our files by loading them as remote files. So I'm going to click on this link here and go into the web browser to show you 3 of the files that I've already preloaded for you. So this is the viewer that Peter was showing you, but now I'm sharing with you a preloaded set of files from the treatments that we just did an analysis from. So I picked 1 file from day 0, 1 file from day 5, and 1 file from day 7. This was one of the NAC domain containing proteins. And as you can see, there is a difference in what the BAM outputs for each of these treatments. So it's a little bit deceptive, but you see little numbers here because these viewers are adaptive. So they adjust the height to like the amount of data that you have in your BAM file. But in order to actually know the true number, you kind of have to look here. So you've got, 2, it's a log2 scale, 2 reads on average aligned, 100 reads on average aligned, and 42 reads on average aligned, to the same reference genome, but on day 0, 5, and 7. So you can see there's a little bit of an up regulation, a little bit of a down regulation. If I want to see the tracks, I can just go here from adaptive view to show all. Okay, and there are no tracks for that one, right? It only had 1 or 2, but you'll see here, there's a lot of cool things and features like built into Genome Viewer. So we have lots of webinars on those also, so I'm not going to like belabor it, but you can kind of see here that by looking at all the tracks, I can kind of distinguish that there's a lot going on day 5 that wasn't happening on day 0. So that's kind of the beauty of doing an analysis from a notebook and then straight into the viewer. It just saves you time, and then you're not having to download a lot of stuff to your computer directly. You can just kind of stream it all. And then if you want to add another track here, it's simply a matter, we already kind of gave you publicly available tracks so that it would be less lift for people who aren't really familiar with this technology before, but just to give you like a little bit of a taste for it. But you could certainly generate your own BAMs and indexed BAMs and BAM indexes, and then put them into your GitHub, and then just kind of copy and paste them over from the GitHub. So I give you the format down there on how to do that if you don't have an FTP site. But just it's really easy because we have the FTP site set up, and these files remain public to you. We can add another remote file from another sample here, and we add it. The trick is you have to have the BAM file and the BAM indexed file in the same directory for it to recognize and upload it. So you can see the new one I loaded comes up at the bottom, right? Yeah, oh no, 37 is the new one. Yeah, so it's right here. So the new one came up here. I'm not sure how that works, but you can reorder, you can move things around. So you can see here that on day 7, in this particular sample, it wasn't that exciting either. So just looking at one sample, as you know, is not enough. So you could probably do some more downstream analysis, looking at counts and averaging counts across the different treatments, so they get a really true view of what's going on. But if you're just interested to know whether this gene's being expressed in your genome in a particular RNA-Seq data set, this is a notebook that will help you accomplish that. Okay, and that's kind of all I had to say about that. And yeah. Peter, how do I hand it back to you?
So don't hand it over to me yet, because there's a question that I think is best answered by just showing them in the notebook. So one question was, what's all the software that you needed? And so I think you made a list of that at the top of the notebook, right? So on what your requirements were for that. So if you could just explain that to them.
I put it here with the install instructions linked.
Okay, and just in terms of the notebook, and I'll show you a slide in a minute that has the link on it too. And the notebook is the notebook file itself is in that FTP directory that I sent to you, including and also the slides that I presented. Also, that's where all those BAMs are and the index files that Adelaide was loading. The other thing is that you can run this same notebook, if I'm correct in understanding this, by going to Binder and running it yourself there. Correct, Adelaide?
Yes, this is all on Binder. This is a publicly available live notebook that all you need is a web browser to get to. So we do provide the link to that in our presentation slides.
And I'll switch back to those now just to show you all those links. So this is a set of websites here that we talked about today, but that compressed URL right there in that last slide will take you to that Jupyter Notebook on Binder where you could run it yourself if you wanted to. And then here's some links to things about, I've already covered some of these in the slides. Another question, Adelaide, that I'll let you help me answer. You know, to run the Jupyter Notebook, you need some kind of environment to run it. So, for example, if you were going to run this on Windows, you'd need something like Anaconda, correct?
Yeah, they would have to set up a notebook on the machine that they want to run it on. So yeah, that would require some more installs and things like that. That's why I made the Binder available, because for people who don't know how to do that stuff, they can still run the notebook. Or like I said, you can just copy the commands and take off the exclamation point and run them straight from terminal, and they should work if you've installed all the software.
Right, and so in that case, you could install all this software on either a Linux or a Windows machine. You just have to go through the steps of installing it. But to have the Jupyter Notebook environment, you'd have to have some kind of environment that will run that. And there are programs that install on Windows that'll do that, and Anaconda is one of them. Another question that I'll answer, and Wayne actually answered this too. Somebody pointed out, I think they know about Magic Blast, but in order to, Adelaide had used FastQDump to actually bring the data locally. That was basically just to show you that you could do that, because you may need to use those FastQ files in some other application. But it is true that Magic Blast can actually act as a client and go to NCBI, or actually, I think if it's in the cloud, it knows that and it will get them from the cloud bucket. But so all you have to do is give it the accessions directly, and it'll run it that way too. So that's right, that's another way to run it. If your only purpose was to run Magic Blast, that might be an easier thing to do. Just the point was to try to show you that you can use the SRA toolkit to bring that data locally. Okay, and I think we've answered all the questions that we had in there. I think that's a wrap, everybody. Thank you for coming, and I'll go ahead and end the webinar for everybody. Thanks, Adelaide, and thanks, Wayne, and we'll talk to you next time.
Thank you, Peter.